Hello class and welcome to our third video. Today we are looking at topic 1.3 and that is genes. As always, we have our subject outline that we are linking this to. So by the end of it, we should be able to distinguish between exons and introns as coding and non-coding segments of DNA that is found in the genes in eukaryotes. We should be able to describe how both exons and introns are transcribed, but only the information contained in exons is translated to form a polypeptide in eukaryotes. Notice how both of those are mentioned in eukaryotes. That should be the main focus of this video. Jumping straight into it, bit of background again, you'll get sick of hearing it, but each time we go over these videos, we're adding a bit more depth to our knowledge. Living organisms have DNA. We have a firm understanding of this by now. And this DNA is not directly involved in running cell operations. It is actually the synthesis of macromolecules, ribonucleic acid, and proteins. This is what DNA directs. So those genes, they code for our RNA, which helps make our proteins. These are the macromolecules that carry out the cell's biological function. And let's jump straight into it with our RNA. So RNA is a nucleic acid, and as such, we should expect it to be a little similar to DNA. We can see in the picture here, we have our DNA on the right, deoxyribonucleic acid. On the left, we have our RNA, our ribonucleic acid. We can see the bases present on the right for the DNA. We can also see the bases present for the RNA on the left. Like DNA, it does have a sugar phosphate backbone. However, it is only one strand. So because of that, it does not have the helix-like shape. This image is showing it. Typically, it would not. The bases it does have in common with DNA are cytosine, guanine, and adenine. However, we get to the fourth one and they are different. So unlike DNA, it is single-stranded, there is no double helix, and it contains ribose rather than deoxyribose. The fourth base in RNA is uracil, and it is swapped out for thymine, and there are different types of RNA. Just to point out the difference between the thymine and the uracil, we can see that on the functional molecule, we have CH3. This is a methyl group, whereas on the uracil, it does not. Otherwise, they look very similar. The reason uracil is in RNA is because it takes more energy to make thymine than what it does uracil. So it can be easily disposed of. However, this does mean it will degrade quickly. RNA molecules are also typically smaller than DNA. RNA molecules do not copy the entire chromosome. They copy genes. So just to compare the deoxyribose to the ribose, we can see that they look very, very similar in terms of molecule. However, the difference is on the deoxyribose, we have a hydrogen located here, whereas on the ribose, we have an OH. So because of that addition of the oxygen, it is less stable overall than what deoxyribose is. So this aids in the nature of the molecules, making them break down quicker so that they can be used for other processes in the body. The three types of mRNA that we will see is mRNA, which is our messenger RNA, our rRNA, our ribosomal RNA, and we'll go over this more in later videos, and our transfer RNA, our tRNA. Some of these may be familiar from Year 11 biology. And just to give you a quick overview of what each of those RNAs does, is the mRNA encodes for proteins, the tRNA carries amino acids, and the rRNA forms the ribosome. Moving back onto genes. So DNA molecules are arranged into one or more chromosomes. Humans, I should say, how many do we have? And you should be able to say exactly how many pairs a typical healthy human should have. So a gene is a section of this chromosome. Within a chromosome, you will get hundreds to thousands of these genes. These are the nucleotide sequences that code for proteins. Proteins are long chains of amino acids that are folded into shapes. This shape gives them their structure and their function. When we are referring to genes, they are the genetic information that is inherited and can code for your traits. DNA is inherited by living things. We all know that it comes from our parents, whatever those organisms might be, and this is referred to as our genotype. Our genetic code is our genotype. This leads to specific physical and behavioral traits, and these are known as your phenotype. So whether you have brown hair, brown eyes, these are the physical expression of those genes. We can see them and we characterize them as a phenotype. The process by which DNA protein directs RNA and protein synthesis is known as gene expression. So the gene expression is when we are making the RNA and protein synthesis. It does this through transcription and translation. More in depth in this in later videos. Today, the highlight should be our exons and our introns. To get there though, we do have to understand a little bit. So first, transcription. The DNA nucleotide sequence is rewritten into complementary RNA nucleotides. What is that? So going back to the earlier slides, we saw that they had very similar bases. We have our DNA in our original strands. It gets opened up so that the RNA can be copied, replacing where T would be on an RNA molecule with uracil, so U, so A to T. However, with the DNA having A present here, it must be a U on the RNA. G to C, T to A to U again, G to C, and A to U. This being our RNA. The resulting RNA is a reliable transcript, and this is because of our complementary pairing. It can't read one of those bases and go, I'm just going to put a random one in. It must be its counterpart. What is first made is known as messenger RNA. So this first sequence that gets made is our mRNA. This carries the genetic message from genes to the protein synthesis machinery known as ribosomes. Moving on to translation. The mRNA is translated into amino acid sequences. Again, this is just an overview. We do go into more depth. Please don't stress if you don't know all of this yet. We will learn. It forms a polypeptide. The polypeptide is what will make up our protein. So it gets folded into different shapes, and based on that shape will dictate how that protein can function. The sites of translation are ribosomes. This is where they get made into the polypeptides. Macromolecular complexes that link the amino acids together are found in the cytoplasm. So what we have here is we should know that we have our original DNA strand. We undergo transcription to make our mRNA transcript, shown in red. We can see the difference between these with the uracil that is present. Note that this is something that you should be looking for. If it is RNA, you should be able to see uracil somewhere, especially in mRNA. After we have our mRNA, we can then translate it into our polypeptide, which is our chain of different amino acids. Very briefly on prokaryotes. Prokaryotes do not have a nucleus. So our eukaryote on the right, we can see in purple the nucleus. We do not have that in our prokaryote. we just have the DNA as a nice big loop going through the cytoplasm, or we should know from previous videos, the nucleoid region. So because it does not have a nucleus to separate transcription and translation, they happen almost simultaneously. So whilst transcription begins, translation can almost happen at the exact same time, making proteins at a much quicker rate than what eukaryotes can. This is essential to their survival and why they can reproduce quite quickly. However, when we're looking at our eukaryotes, they do have a nucleus. And because they have a nucleus, this separates transcription and translation. So transcription happens in the nucleus. Everything in transcription happens here. Everything for translation will be outside of the nucleus. Something new to introduce is that we now have pre-mRNA. This pre-mRNA is made of a complementary nucleotide sequence to its genes. So that pre-mRNA, if we go back to previous slides, this would be our pre-mRNA. It is a direct copy of the DNA strand with everything that it would have paired to. So this pre-mRNA does mature into, well, mature mRNA. Often when we refer to mRNA, it is the mature version that people typically refer to. If they're talking about pre-mRNA, they will use pre as the suffix. So both ends of pre-mRNA are modified and specific interior sections are cut out. That is what we're building to. And the resultant spliced together molecule is that mature mRNA. So that direct copy of the genes for our pre-mRNA has sections in it that we don't want or we don't need. Our protein might only code for the light pink colors here. The mRNA is then transported into the cytoplasm where it has access to ribosomes so that it can undergo translation. Okay, moving on to the RNA processing. So pre-mRNA is the same length as its gene copy. Whatever that gene it copied is, it is the exact same length, replacing thymine for uracil. Enzymes in eukaryotes nucleus modify this pre-mRNA. The ends of the pre-mRNA have chemical ends added to facilitate this export as well. This prevents the degradation. As mentioned before, uracil can degrade quicker than what thymine would. These ends also help to attach to the ribosomes. One or more portions of the pre-mRNA are moved and the remaining are reconnected. This is known as RNA splicing. So if we're looking at the picture here, we see our full pre-mRNA, the copy of the gene, and we have something called exons and introns. Just from looking at this picture, we can see that to go from this pre-mRNA, something happens, splicing, and what we're left with is the mature mRNA, and just by this picture we can see that it must be made up of these exons, because these are the three components that have been attached to our mature mRNA. Sorry. And what gets taken out is our introns. We don't need them. So, as an example, pre-mRNA could have up to 28,000 nucleotides. That's a copy of the gene. The mature mRNA might only need 1,400 nucleotides. So long stretches often contain non-coding nucleotides, and that is what we refer to as our introns. They're non-coding, we do not need them, they can go away. We just want our exons. So the coding segments that make our proteins are known as exons. The final part is how those exons are put together and how the introns are removed. The removal of the introns is done by something known as a spliceosome. So it's a few little enzymes that are put together which make a large complex protein and RNA. This binds to several short nucleotide sequences. So we can see here if we have our exon, our intron, our exon, we have our protein and other proteins. It's not just one, it's actually a combination which make our spliceosome. We can see that it's attaching to either ends of those exons, bringing in the intron to make a loop. Once they're really close together, it will detach one side of it, so we'll cut it off, bring it in closer, reattach the intron as it also disconnects it from the other side and simultaneously join up the exons. So what we get is we get this little loop that comes in, gets joined really close together, gets severed and cleaved, so it's cut off. The intron is free to go. We are left with exons that get joined together, going from our pre-mRNA to our mature mRNA. The intron does degrade quickly, and this spliceosome, as I said, joins the exons onto either side. So what we have now is a crucial sequence of introns in eukaryotes. A single gene can encode more than one kind of polypeptide. So polypeptide are our proteins. What we have is an entire gene that can make more than just one polypeptide. Depending on which sections are treated as exons, so here we have 5 different exons, depending on which ones get put together, we will get something known as alternative splicing, or alternative RNA splicing. So the way that these get put together will make different proteins. Some other examples are we have our gene, we have our exon, intron, exon, intron, exon. Introns can go away, don't need them. But what you might find is that different exons will code for a different section or domain of the protein. So if this is our protein molecule, to one big piece, we have different exons that code for different domains. Just think of these as sections of a protein, and often they will code for different components rather than making just one long protein. So different exons can often code for different domains of the same protein. Alternative splicing allows for the number of proteins to be much greater than its number of genes. So just because we have our gene, let's go back to this. Just because we have our one gene from our DNA strand, we can get multiple proteins because of this. And this is why humans can carry out more complex functions than other species with the same number of genes. All right, or as I should say, all right. What you needed to take away from that is a gene consists of a unique sequence of nucleotides that codes for a functional protein or an RNA molecule. We can distinguish the difference between exons and introns as coding and non-coding segments of DNA found in genes in eukaryotes, and describe how both exons and introns are transcribed, but only the information contained in the exons is translated to form a polypeptide in eukaryotes. So transcribe means it can be copied, but once it is translated to make our protein, it is only the exons that are present. We'll go over more in class. Thank you for watching the video.